And welcome to the Evidence Reasons Academy, the sister channel of the Evidence and Reasons for the Christian Faith video channel. This is where nerds hang out, Christian nerds, and we cover a lot of academic topics that are kind of important to understand, especially the creation evolution controversy. We cover all sorts of things, history, economics, physics. Oh my goodness, I've been just enjoying all my physics stuff. Even though hardly anyone else even would watch it. I've had more fun doing the physics stuff. I get 10 views and I was so proud of just having only 10. I'm not really eager for big channels. Today, we have a lot to celebrate. Emory is getting inching closer to graduation, and he's passing through milestones and exams and doing well. So do you think we'll have a Christmas present, an early Christmas present? God willing,
Lord willing. The final draft of my final paper is due, I think, on the 8th. So, and I do not expect that there will be any difficulties with it. My professor has already approved it in principle. He's just making edits, or requesting edits. So I expect that I should be done as of the 8th of December. So, Lord willing, fingers crossed, I should be done by then.
So, you're kind of our ploidy expert, our resident ploidy expert, and
bit of a nerd, more like. But anyway, go ahead.
Okay, as far as the ploids go, haploid is my favorite because it's so easy to model.
Yes, the math is a lot easier with a haploid than it is with a tetraploid or a octoploid. I've tried to do so. I decided one time I was going to try this just for the sake of destroying every brain cell in my body. That I was going to try to model out a Punnett square for a tetraploid with a dihybrid cross tetraploid, and oh my goodness, the math! Oh! I have a copy somewhere of that, but it hurt my brain so bad to do that. Oh,
okay. So I have a confession to make. You know that I'm. There are not many creationist population genetics guys, and it's kind of scary that I'm kind of in that handful of the few, and I don't know that much. And I really love haploid models because even going to diploid starts to be nasty.
Yeah, you get beyond a single trait cross in diploid, and you're getting into dicey math, and you try doing something along the lines of linkage disequilibrium, and then the math starts getting all kinds of fun.
So, how this conversation, how I said how I got interested in this. There was a paper by Michael Lynch that I was reading. I was doing kind of a nerds go clubbing thing, and I didn't even get through half of it. They're talking about multimeric proteins. I've been hammering on that because evolution in multimeric proteins, I'm just like, okay, if it's critical to function, this is a disaster for evolutionary biology. And people are like, they don't even understand what I'm trying to say. So maybe, I mean, there are times, honestly, I think sometimes my arguments are a little too subtle for people to understand. And I get that. And I'm sorry to be mean. I'm kind of in a mean mood. Critical thinking and evolutionary biology don't mix. I mean,
no, they don't.
And it's like, okay, so I showed this this off repeated graph, and I'm gonna show it here in the I'll put it up on the screen in a little bit. Hang on, let me get all my buttons and all my switches in order here. Let's see. So I'll share the screen. See if I can get this photo. It's maybe a little slow in loading. Man, it's really slow in loading.
Is this the photo you sent me, Sal? Because if you need me to, I can try to share it. So, do you want me to try to share mine?
Um,
Is this what you sent me, the paralog ortholog thing?
no,
Oh, okay, never
I
mind.
don't know why this thing is. This is just being just obnoxiously slow. Let me try to kill it. Oh yeah, it hung.
Wonderful.
So let me just kill the system. It's dead. So I'll go to cross circuit B. Just that's just a joke. All right. Okay. Now I'm getting all these messages that it locked up. It's like, yeah, no kidding. So
Broken.
yeah. So let's try this again. All right. I found an alternate way to render it. So, I mean, you've seen this before. So it was funny. So I was saying, okay, morphologically, it's so obvious. I mean, in the molecular shapes will tell you there's not intermediates, there's discontinuities. That's why I love this diagram. And then they're obvious discontinuities as let me see if I could find it here. There are discontinuities also in sequence. Now, I specifically picked out these examples because you can actually visually see the problem. And it kind of shocked Aaron Raw. He said, "What about that?" He said, and I said, "Why don't you ask Dr. Dan?" And he came back and he wrote me an angry letter and he said, "There's no effing common ancestor for proteins." I was like, "Well, thank you for admitting my point." And he said, "That doesn't mean anything." I'm like, "No, I don't think you understand the problem, sir."
Yeah. And if I can just maybe put the problem very simply, and I know we're on the academic channel, so it's a little maybe not to be quite so simple, but effectively the problem is if there's no common ancestor for proteins, that means every protein family had to, or maybe not every protein family, but a lot of the protein families had to arise independently. But protein families are shared across a wide range of ancestors, which means a wide range of groups, which means they had to be phylogenetically early, which effectively means that you had to have multiple protein families arising in effectively one common ancestor, maybe a couple of common ancestors, a couple of ancestral places. The problem with that is that you have to get a bunch of protein families arising in the same species or group of species relatively rapidly, and then diversifying across lineages for however long it takes. That's not trivial.
Yeah, that means when you had the eukaryotic transition with all the critical proteins, only one eukaryote survived and had to go through that one little cell. I mean, okay. So you have these little pools of cells. But the other problem that I think is more daunting, and like I said, evolutionary biology and critical thinking don't mix. It's like, guys, do you not know anything about protein biology? This is so difficult to get it to be functional. I mean, even the ones that do have even less excuse. So look at this potassium ion channel. The hole in there has to be less fractional angstroms. I mean, this is less than the radius of a sodium atom. That if you're off by that, this isn't going to work. You misspell this, it's going to fail. And this is a multimeric protein, meaning
And let me before you go any further, Sal, let me explain what a potassium ion channel is, so that anybody can understand why this is important. Potassium ion channels are in the cellular membranes. They transport potassium across the cell membrane. You don't have these things, you die.
you die. And I was, Dr. Dan said, did you figure in natural selection? I said, yes, you're dead. If it's not there, you can't evolve something that's dead. I repeated it twice. And he said, if you don't factor in natural selection, I'm not interested. Well, and I want it, I'm trying to be nice to him, but sometimes I have to just point it, I'm going to have to be a little firm and say, sir, do evolutionary biologists calculate the probability of selection? They don't. You'll accept that, but you won't. I'll give you good reasons that my figure is correct, and you'll say you're not interested. And here we have Janet Duttridge. She says, leaving but wanted to give my support. Let me say a quick prayer for you, Janet. Dear Lord, Janet is sometimes almost comatose every day because of her health issues. She's not able to digest food well and process it, and she has all sorts of issues, Lord. I thank you for the loving husband she had that was with her until he passed away, and she'll meet him in heaven. Please comfort her while she's here on earth. And you, Lord, we know that you care for widows and orphans and the disabled. And Janet is disabled as well. So please comfort her, help her to rest, give her strength, and give her family support that will love her and provide for her. In Jesus' name, amen. I got to tell people, when I see how much Janet carries on with her Christian life, it's given me a lot of motivation to carry on. I said, if she can do this, I can do this too. Yeah. So thank you for your courage and example. You've been a comfort and strength to me. It reminded me of my mom, when all she could do most of the day is to sit in a recliner and spend many hours in prayer. And and she, that kindness and that perseverance inspired me, who is able-bodied, to just persevere. So thank you, Janet. I want you to know you're special to the Christian community. When it says in the Bible, when I'm weak, I'm strong, and God is working through you. So back to the nerd session, and thank you for joining us, Janet. So, yes, you're dead. And I just like Dr. Dan.
Yeah. And the problem with a selectionist argument for this is there's actually several problems, but the first problem is it's an all or nothing solution. If you don't have this thing, the cell dies. I mean, there may be a few cells somewhere that don't need it, but most cells need this or they die. It's literally that simple. But,
okay,
go ahead.
Can you hold out your other thoughts?
That's fine. I got the rest of
them. Yeah, I just wanted to elaborate on it dies. I'll tell you, I was writing Aaron Raw on this, went over his head. And because Cindy Lincoln was in the email chain, I just wanted to show her. I said, don't let Aaron pretend he can bully us. Mm-hmm. He doesn't know what he's talking about. Yeah. And he said, the way I define this, he is incomprehensible, calling me all sorts of names. But the funny thing was, I did a little research. They said, how is it that cells have potassium ions in them? They said the origin of life, we have to solve this problem for the origin of life because it has this intense potassium ion concentration, so critical. Mm-hmm. Yeah. So they came up with the idea that the first life must have evolved between mica sheets where there was plenty of potassium.
I mean... I've heard more ridiculous ideas than that. Like the octopus being put here by space aliens, and that's actually a legitimate thing that was published in a peer-reviewed journal. I'm not making that up. They actually published that. But that's reaching pretty hard right there. I don't know that much about mica, but I wouldn't think mica sheets would be a good place for life to hang out. Like, I'm not an expert on mica, but
Well, yeah. I mean, what happens if you move outside of the mica sheet so you can get sunlight or some other energy source? You lose the I mean, it was critically dependent on 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 maintaining a potassium concentration. I'm just like, yeah, so how are you going to evolve a potassium ion channel? And then, so I cut you off because I wanted to tell you that. That's the only reason I had to cut you off and interrupt. But you had some other problems you wanted to point out.
Even from a selectionist perspective, selection doesn't operate on. Oh, it operates at the population level, and the effects are expressed organismically. So even if we supposedly developed a cell that happened to get lucky and get a giant mutation that gave it a potassium ion channel, which, by the way, Sal, how long is that protein? Just do you know off the top of your head how long that protein is?
About 1,000, but there's some complications.
Okay, so we're looking at 1,000 amino acids approximate, with complications, obviously, and it has to fold a specific way.
Not that. Remember, it's tetrameric. It has to have what they call a translocation process. It has to have code so the translocon machinery can actually stitch it into the membrane. You can't just, it doesn't pop out of the ribosome and get in there. You need to shove it into the membrane,
right.
Stitching, and there's a whole ritual that has to be just right.
Yep.
So and then not only that, you need to have the right. I think you need the hydrophobic on the inside and the hydrophilic on the outside.
You need the hydrophobic piece needs to be in the middle, and the hydrophilic ends need to be on the outside. Yep.
And then so that stitching process, it needs well located codes, not just one.
Yeah.
They have to be positioned so that the stitching machine can put it right. And it's like, good luck. What are the odds of you getting that? See, this is why I like studying proteins, because, for like 20 years, all we had was citing Dugac's figure of 10 to the minus 77th power. We've since understood now, and Joe DeWeese and others, Kirk Durston will back me on this, every protein has a different set of probability arguments just because of just like the requirements needed to be met, how difficult you have to hit the target. I mean, even just to get into the into the membrane, we're not even talking about a large amount of any functionality. Just being able to stitch it in the membrane is not trivial.
Yep. But let's just ignore all of that, which that's 5 minutes of ignoring we have to do, of problems we have to ignore. But even if we ignore all of that, a 1,000 amino acid genetic code is 3,000 base pairs. But except it's more than 3,000 base pairs, because there's something called introns, which go between the exons of every protein domain. So we're looking, in order to make this, we're probably looking at 4,000 to 5,000 base pairs to make this gene.
For bacteria, we don't have to worry about that. So we presume, let's just be,
even some no, that's right. You're bacteria. You're okay. Bacteria. So for bacteria, we'll say that we'll just go back and say we have a complete just gene, no introns. We don't have to worry about that. So we'll give it 3,000 genes, although that's disputed because introns' early hypothesis says that introns existed in bacteria and then they lost them later. So,
introns are.
So no, seriously, that's a thing. Like, I'm literally staring at my notes from my genetics class. Like, that's a thing. So depending on which view you take of that, but we'll give them the benefit of the doubt and say that's 3,000 base pairs. Now, I don't know how similar this is to other proteins, but I would suspect that that's going to be more than one mutation to change that into a potassium ion channel. I suspect you're probably going to need dozens, if not hundreds, of targeted mutations to change this thing into a functional potassium ion channel. And that ain't happening on a 3,000 amino acid sequence, 3,000 nucleotide sequence. You're not getting one mutation where you want it is 1 to the 3,000. Getting a couple dozen, your numbers are impossible.
Yep. You mean like I think it would be more like 4 to the 3,000th power. Sorry, just to be you know, I'm
that's
going
true.
to
You're right, because it would be you had to get the correct amino acid. It's not just one change. You have to get the correct amino acid into the place. So it's actually worse because you don't just have to get a change. You have to get a specific change out of 4 options.
So, I mean, while we're at it, did I have? Okay, so there are a variety of videos on this, but one I have a copyright release on, thankfully. But unfortunately, it is of the endoplasmic reticulum, not the cell membrane. But it's the same. It's the same principle. If you have 4 minutes, I'd like to show it just to kind of deliver it. Because there's an evolutionary biologist saying this is not a problem for evolution. And he said, I said, empirically and theoretically, you've got challenges from physics, chemistry, statistics. Mm-hmm. And I believe he's a senior person. I could tell also that I think he's important enough that I should at least try. And at the very least, it actually gives me a chance to do batting practice, to make sure my arguments are good in case he has some data points I need to be aware of. But this translocation process, I'm just like, okay, this is telling you what the constraints are just to get it in the membrane. And remember, we only have one shot at this. Mm-hmm. So even if you co-opt something, you better co-opt it 100% right, and then you have to modify the parts. Mm-hmm. I'm sorry. I live near a fire station, so sorry for all the extraneous noise. And I'm trying to get through the commercials here.
Don't worry.
All right, so just as a qualifier. So just as a qualifier here, okay, this is not the cell membrane, but it's still the same principles of inserting in a cell membrane. This is the endoplasmic. The membrane is the membrane in the endoplasmic reticulum.
Yeah. So that's the cell membrane and the endoplasmic reticulum membrane are functionally identical. They're both made of the same type of thing. So getting it to insert into the endoplasmic reticulum membrane is going to be the same as inserting into the cellular membrane, or at least very similar.
Yes, that's the point. But this happened to be one of the better videos on it, and that's why I'm sharing it. So I have the Chrome tab, 15.4 translocation. All right, so I'm going to mute our microphones, and then we'll watch this.
Or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are 2 separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. 2 kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling a sewing machine.
Like, guys, can you look at these? I mean, these guys tell me with a straight face that's not a problem for evolution. I'm just like,
Okay, there's this. Or
Isn't that cool?
Yeah. You guys need to explain how selection causes that.
Yep.
I can maybe buy that selection can operate at organismic levels, and even some of that is up for debate depending on what we're talking about. But at that level,
Yeah. And, even just dip a little bit into the origin of life thing. This whole thing when Shostak came out and say, we're able to create a cell membrane. I'm like, no, you weren't. You're able to make the equivalent of a soap bubble because you would have to have all that machinery to make it. It's like, just because you can make this little spherical thing, why do you think all the rest of this would be there? That was pretty illegitimate. And Godzilla Freak is here. If it just so this goes to Michael Lynch's paper. So let me see if I could put this. I'm going to stop that share of that video. Thank you for watching that with me. Because sometimes I got to confess something, and I realized this. Our social interactions can affect what we believe is true or false.
Mm-hmm.
So much so it can even override our rational faculties. I mean, for a kid, that's really important because he thinks, well, this feels good. Why is mommy and daddy making me do this? So it's good that he can be socialized to do certain things that he doesn't understand.
Good and bad, depending on who's doing the socializing.
Yes.
Groomer schools.
Yeah, unfortunately. So why did I bring that up? I got to tell you, sometimes when I'm around people that are so stubborn like that, I begin to think, gee, do I have it wrong? I mean, seriously. I mean, I thought I just blew apart Aaron Ra's arguments, and he just kept being like a stone. It's like didn't budge. And like, is it me or is it him? Of course, now realize it's him.
Yeah.
But you begin to have a little bit of self-doubt when you're around someone that confident. But then after that, after you've had a few interactions, it's like, nah, I'll never trust him for anything, because I just assume he's wrong.
Yeah.
And I just stopped. He kept sending really long emails, and I just stopped. I just kept it. It wasn't worth my time to read it. But what I was trying to point out is that there are two things. One, that's why it's really good for me to hang out with you, because you make me feel sane. Because
most people say I make them feel insane, so I don't know. Maybe we got problems here, Sal.
Yeah. I mean, if you're in such a negative environment, it could start to wear on you, honestly. Mm-hmm. I don't, maybe in my younger days I had, I was so reckless that I would go through graduate school and say, I'm right, I'm right,
Mm-hmm.
and I'm right about young earth creationism. But, as I got older, I got a little bit more tentative. And so seriously, it's great to hang out with someone like you and other creationists, because I found one really disturbing study why transgenderism is spreading among females.
Social contagion.
Yeah. What did you call it?
Social contagion.
And so what happens is, if it starts to be like the cool faddish thing, not because it was a rational thing to do, it's just they grab it because it makes them feel cool and faddish, and they don't realize this has devastating consequences if you really take it to where you're modifying your own body. Oh my goodness.
Even if all you do is take hormones, it has devastating consequences. I mean, it has devastating consequences even if you don't take hormones, but the consequences of you taking hormones are then maybe disfiguring yourself permanently because of your desire to be something God didn't make you to be. So it
ends up being, and where this really hit me was, they said, okay, let's take it to something less charged, something less controversial, anorexics.
Mm-hmm.
We've also had contagion, a social contagion with anorexia and bulimia. Since the advent of social media, we've known, and this is Abigail Shire, she did a study and she was on Jordan Peterson's show.
Yep.
And it's really great to hear professionals saying, we're trying to help people that are having psychological issues. So we're not going to be politically correct. We have to be responsible to care for our patients. And Jordan Peters is saying, the American Psychological Association, they said they're threatening to de-license him for speaking his conscience. And he said, I know this is terrible, and I can't say it to my clients without losing my license. I don't remember what the resolution of that was. It might have been, he said, go ahead and take it. I'm going to tell the truth. I would expect that from him. And so what Abigail Shire pointed out, she said that, okay, when you have anorexics in a hospital ward, you try to keep them separated because they'll reinforce each other. So here it is, the girl's starving to death. She's skinny thin. You can present her the scales. You can put her in front of a mirror, but she says, I'm fat.
Mm-hmm.
And if she's with another anorexic, it's just going to reinforce that.
Mm-hmm.
Women are especially vulnerable to that sort of socialization of their.
Yes. Now we're getting into things that you really can't say, like that men and women don't react to the same things the same.
Yeah. So, even me as a guy, I'm kind of like, I didn't, not that I had the greatest respect for Aaron Ra's intellect. Even then I was getting a little affected, And I say, am I this clueless? And then either
you
that
don't
or
have
he's
to have a respect for his intellect because he comes across very confident. I think that's not so much. It's not so much that you think he's particularly intelligent or particularly well read. He's neither. It's more that he has a Dunning-Kruger level of confidence in his own opinions.
Yes.
And he pursues them aggressively. And if you're someone who is a little bit more intellectual and is actually trying to figure out what the truth is, someone like that can be very persuasive because they unlike you, they are confident that they're right. And normal intellectuals, people who are a little more intellectual, tend to be a little bit less confident that they're right, and they also tend to carry a little bit of imposter syndrome. Like, what if I'm really stupid and everybody else around me is really smart? And this is not just applying to you, Sal. This is a general observation. Someone like Ra, who is belligerently confident in his views, can sound very persuasive because the people who are a little more academic are like, well, I'm not really sure about what I think, but he sounds really sure, so maybe he's right.
Yes. So at a neurological level, that's what was happening. And so I looked at this and I said, no, I'm right.
Mm-hmm.
And so spending time with you and seeing other people saying this is just incredible. It's just like, yeah, me as an engineer, I could work, I could see it. And then, but then evolutionary biologists said, we're trying to fix you, Sal. I don't think you really understand. And it does get to you at some point. But on the other hand, I would dig in my heels and say, okay, if you're that confident, show me the numbers. Show me the math. I can do the math. Do you have any? Do you even try? And maths is here. Social contagion sounds a lot like lactose intolerance. It's like we're assuming men are the normal ones and females are behaving abnormally. Lactose intolerant people are genetically normal.
I mean, he has a point about the lactose intolerance. So that's well, to an extent, because all humans are born normal humans are born lactose tolerant and then lose it over time. Because if you weren't lactose tolerant, you couldn't take your mother's milk. But over time, that's lost. The people who don't lose it are the abnormal people. So yes, you're right. You're right. Those of us who can eat ice cream and love it, we're abnormal. I grant you that. But when I say social contagion, I'm not saying just applying that to women. I'm applying that to children as a general thing. Now, women tend to be a little bit more susceptible, maybe because women and men respond differently, and women tend to respond more emotionally, and men tend to respond more rationally. And there's nothing wrong with either response. It's just they're different. And they balance each other. By the way, this is why marriage works, because men and women respond differently, so they balance each other. But when I say it's a social contagion, what I'm saying is that children are very susceptible to the ideas of their peers. This is why the idea of peer pressure works. And if something is being portrayed as hip, cool, and it will get them positive attention, many of times they're not getting positive attention at home. Either they don't have a father in their lives, or maybe they don't even have parents at all, because our societal fabric is falling apart. If they're getting positive attention for self-identifying as gay or bisexual or transgender or fill in the blank, then yeah, they're going to do that. I mean, I was reading a story yesterday from a mother in Seattle whose daughter socially transitioned at school, and her parents didn't find out about it until her therapist called them and said, hey, your daughter is actually a boy, and I'm going to give and I want to meet with you to discuss how she can transition it at your house. I'm giving you 3 days to adjust to that.
It's terrible.
Yeah. And apparently this poor girl, because she had socially transitioned at school, she was going on some school camping trip or something like that. And her teacher, who apparently was grooming her to a certain extent to do this, suggested that she stay in the boys' cabin. She's in 5th grade. And she thought she couldn't say no even though she was really uncomfortable and didn't want to do that because she had already told everybody she was a boy. You see how this works?
Yeah. Hideous. And
that's what I mean by social contagion.
Contagion. Well, unfortunately, and I'm learning these hard lessons like in the last 2 years, I'm realizing that's how cults and religions are perpetuated outside of evidence.
Mm-hmm. A lot of them are.
And,
you know, kind of. Do you think everyone in Saudi Arabia wants to be a Muslim?
Yeah. But I mean, it's very sobering how much people can surrender their faculties with, you know, someone so, you know, the social pressure and it's, you would think it would spill over into the sciences, but evolutionary biology is just driven by that. I'm just like, do you guys not see the emperor has no clothes? And it's like, yeah, I guess they don't. And same for the abiogenesis community. Until someone starts calling it out.
And when somebody starts calling it out, the first thing they do is double down.
Yep. And so, you know, I mean, it's good always double checking because there are times I do find out I can make my argument stronger. I had a weak point there. So when I review it, you know, now it's just like, no, you're not going to win this because you don't have the numbers. The numbers don't add up, Aaron. And then he started making more non sequitur stuff. But I began to think, why was he wrong? I said, I'm not going to do kind of like what's typical on the internet where you just kind of just try to BS your way out of it. I said, no, I'm going to really try to methodically think through this. And I may point out his flaw in reasoning. He kept making morphological arguments and he doesn't see the problem. And I said, you know, he's wrong, but, you know, how can I effectively counter? And the thing that I found as I thought about this, and this is slides I've shown you before, but at the morphological level, let's say just, you know, this is a designed evolution, okay? But you could see it's incremental. And if you look at this, it's like, well, you know, they're really not that different. The problem is you won't see the differences when you're just kind of studying the rote morphology. Even like something, let's say a luxury car, it may have computers in it, thousands of transistors. I mean, this is not a trivial change just to add a computer. Actually, modern appliances have multiple computers and operating systems. The technological leap to even have like GPS, say cell phones and other kinds of computers in here, and all the chips, the technological leap is gigantic. And so I was trying to, you know, Aaron doesn't understand. You cannot measure. This is not, looking at changes in morphology, which he's a big taxonomy guy, and just like, dude, this doesn't work because it's not correlate. It's not sufficiently correlated. You can have huge changes at the molecular level. You're not going to know. And that's why I said all of his definitions of macroevolution and speciation, I said, that is subservient. The molecular arguments will take precedence, and it will not explain things like that. You could claim speciation all you want. It's not going to explain changes. And oh boy, yeah, it's not going to explain molecular changes. And he couldn't fathom that. I'm just like, okay, dude, you know, it's like, because he's trying to flip Cindy Lincoln. And I said, okay, Aaron, explain how speciation proves the emergence of a potassium ion channel. Yeah. You know,
it's interesting that he's so dependent on morphology because most of the Evo phylogenies these days have completely abandoned that or are working from a genetic perspective rather than a morphological one. So it's interesting that he's still depending on morphology. I mean, he's almost like he's still in the taxonomical Stone Ages.
Yeah, Dunning-Kruger. just like Dave Farina. Oh my goodness.
That was an epic beatdown.
Epic. You know what? I banned certain people from my channel because they're giving me a hard time and say back up your claims. And I said, well, did you see, James Tour destroyed this? They said, no, he didn't look at this video by Farina. So these people that do that to me, they're automatically banned from my channel. I said, you guys are hopeless. I don't have time for you guys. If you're just going to buy that,
If you think Farina's response to Tour's series of just nuclear holocaust on that video, if you think that Farina's response video to that is anything other than his attempt to cover his mistakes and put some tissue paper on them, I don't know what to tell you, man. I've watched Tour's series. I watched Farina's original video. I watched his follow-up video. Nothing. It's just not there. And no, I'm not necessarily ripping on Farina too hard because Farina's not an expert. I'm not saying Farina. That's not. But he needs to realize that his fans, more importantly, need to realize that Tour is an expert. Shut up and listen to him. And Tour's not even saying that the origin of life couldn't happen. All Tour's saying is we haven't proved that it did.
Yep. And any number of criticisms, but again, this whole thing about there has to be a word for the social contagion that overrides logical. It's just when people can be, well, look, Fetterman was elected over Dr. Oz in Pennsylvania. And it's like, enough said about social contagions. Or Alexandria Ocasio-Cortez winning in a landslide.
Occasional cortex. No, well, let's just put it this way. We now have not one, but two federal officials who are unable to string together a coherent sentence without a teleprompter, and even that's questionable.
I shouldn't laugh, but it's so bad you can't help but laugh. So
my home state's Pennsylvania. I know exactly what happened there. I mean, I don't live there now, but I know exactly what happened. It's very straightforward and simple, but yeah, it just anyway. But people, I'm not so much. It's not even that. It's not got anything to do with those two individuals' political parties. It's more along the lines of they can't string together sentences.
Yeah.
Like I could comment. I could comment on their political things, but that's a whole other kettle of fish. They can't string together sentences coherently.
They're unfit to even be like.
They need medical attention. They shouldn't be in any kind of power. And I'd say that doesn't matter what political party they could be the furthest party right outside of crackpot parties that you could be, and I wouldn't care. Like, I don't care. These people are not healthy enough to be in positions of authority. They're just not healthy, and that it's sad that their families are propping them up for this.
Well, there's some elements propping them up for this. Mm-hmm. The media is covering for them, and yeah, there's some pockets there's some deep pockets lining. Yeah. So, by the way, I'm generally not a drinker, but my neighbor, we were outside, and I just wanted to be a good, just a good neighbor, loving neighbor. So I accepted an invitation. He said, "Come over here, Sal. Let me give you a beer." And so he gave me this. So if you see me turn off the camera, I'm just taking a sip because I can't drink this very fast. I'm not a drinker at all. Yeah. I may have two of these a whole year. So if you see my camera go off, that's just because I didn't want to waste something that was so generously given. And anyway, so again, I just got to say, this is why it's just nice to hang out with you, because I have been affected by this. It gave me a chance to revisit my arguments. So this thing about the multimeric protein, this one, the topoisomerase and the potassium ion channel, the multimeric architecture is absolutely critical. I mean, if we change this somehow, that this changed to three MERS versus 4, it's going to change the size of that hole. So given that this is sensitive to 1 angstrom, less than 1 angstrom, I mean, let me see if I could get the, I'm going to try to bring up a picture of the potassium, sodium ion size. See if I have a picture. I want, okay, here, oh, here's a beautiful picture of how the constraint. Let me bring it up now. You see, so sodium is 1.33 angstroms. This potassium is 1.33 angstroms. Sodium is 0.96. Calcium is 0.99. If we just change a little bit of the size of that fit, it's going to fail. It's going to fail. So why don't I show? Do you have time for a video for the potassium?
Sure.
Okay.
Sure, yeah.
So this is why. Oh, since Dr. Duff is here, he was asking, what's the problem for evolution for the protein orchard? It means that there, at least superficially, there isn't a gradualistic path from a common ancestor. You can't invoke descent with modification. And in the case of something like potassium ion channel, if it wasn't at the origin of life, it had to evolve it. That leads to all sorts of problems, how this can be done in gradual steps. And you may have missed the show, part of the show where we are showing the translocation process and how difficult it is to insert this. Now it's the multimeric problem, and I'm going to show a video. I'm going to show a video on this. Let's see. So potassium. This is the potassium ion channel in a bacteria. So where is it? This is a beautiful. It's this one. It's only it's less than. I should. I'm sorry. It's going through all these silly commercials that to watch. Okay. StreamYard likes Chrome, and so I can't use my ad blocker. That's a different browser. So let's look at this. Let's see. Share screen and share audio. Chrome tab, potassium. Okay, here we go.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. 4 in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
Oh, sorry. So you can see how precise that would be. I mean, there's just okay, and the multimerism is critical to this. There's some proteins. Lynch is arguing there are some proteins where you can have a monomeric to dimeric to like say trimeric, but that may be across species. I don't know if it's in a single species. And I just got to get this here. Next time we're both in Dallas, I'll have to introduce you to Texas whiskey. If you're going to have 2 drinks a year, let it be that and some steak. Oh, thank you. And just for the audience there, I did see Matt's this past January, and he was so kind to take me to a Texas steakhouse and treated me to dinner. Ultra great guy. And thank you for the hospitality. That meant a lot to me. And so perhaps we will meet. There is oh my goodness, there's this Discovery Institute Dallas conference in February. I don't know that I'm going to go. If some people know I have a TV deal for the New Creationist Museum. So maybe on my way to Nashville, go to the Discovery thing, or I'll go to the Discovery thing. And on the way there, I'll go to Nashville, do my TV show, build my museum exhibits. So going back to the potassium ion channel, Lynch's paper was saying, we haven't, why don't I bring up Lynch's paper? Because was, he's really one of the better ones because he has quantitative skills. And there are many times he has moments where he sounds like a creationist. Oh my goodness. So let me find that. And there are going to be some funny things. I was having a good time reading it. I started this 7:45 a.m. last Saturday because I have some sleep issues, and I either sleep too early or too late. That time I slept too early and I woke up and I said, I'm just going to try to do a stream and it might help me go to sleep. So this paper is the evolutionary diversification. All right, let's see. Proceedings, National Academy of Sciences, and I'm going to bring it up fully. And this is where we get to the ploidy part. Evolutionary diversification of multimeric states of proteins. Michael Lynch, June 12, 2013. And why don't I read the abstract, see if I could find the point I wanted. Rather than operating as single units, most proteins assemble as multimers. And we just showed one, the potassium ion channel, usually with all subunits derived from the same gene, in contrast to patterns of gene structure and genome organizations, which typically exhibit substantial increase in complexity from unicellular to multicellular organisms. The structural complexity of orthologous proteins appears roughly constant over the tree of life. Wow. The interfaces of multimers also often shift dramatically over evolutionary time. To explain these observations, a model is presented for the stochastic origin of variation in the multimeric states of proteins via the joint processes of mutation, random genetic drift, and constant directional selection. They assiduously didn't have one for the potassium ion channel or topoisomerase, which are really well studied. Okay, so I got to point out something that made me laugh. Well, he said, although the large pool of oligomeric structures in today's organism cannot possibly be strongly maladaptive, this need not imply that they have arisen by or are currently maintained by adaptive processes. That is a really strange admission, because he's saying, it has to be working. Are we misperceiving this? Is it possible this is maladaptive and we actually think it isn't? So no, that can't be a possibility. That's the way I read it anyway. But this is what's funny. He says, indeed, despite the plausibility of many of the above hypotheses, I think that needs spelling correction. It's implausibility. Empirical evidence for the adaptive value of alternative multimeric structures is essentially completely lacking.
That's a rather stark admission.
Yeah, I told you this was a fun paper. Okay, so now, just to be fair, he did find examples where in some bacteria, like the hexokinase, so what's the color coding here? I think, okay, this dark green is a monomer, blue is a dimer, white is a trimer, 4 is a tetramer, yellow is hexamer, orange octamer. So hexokinase can be in eubacteria in the monomeric or tetrameric form, which is amazing. Archaea, monomeric, dimeric, and tetrameric form, or the monomeric and dimeric form in eukaryotes. And then you could see some that are all monomeric. There are some that flip between monomer, dimer, and octamer and tetramer. So to his credit, you can find some that, depending on the species, the level of components changes. But I have to say again, for something like where what I think in those cases is happening is they can kind of bind together, but it's not critical to their function. You're just going to get a little protein complex that has the enzyme on like 3 or 4 sides instead of one. Yeah. It's not critical to the function. I would wager that these things could probably, if they can work in the monomeric state, if they conglomerate in a certain way, it's just going to amplify it in that complex. That's all that's happening. But for like the potassium ion channel, the number of components is actually critical to the geometry, therefore the function. Same for topoisomerase. So he could have analyzed how this could have happened. And so the probability of forming a topoisomerase, you have to have all what we call the interface points, where you have the charge distribution and it has to be in the right position, and so that it will attract and spontaneously form the structure. It's amazing. We throw topoisomerases into solution, and they'll spontaneously form dimeric complexes. You don't need a chaperone. It's amazing. There's a lot of self-organization just built in by the way of the code. So when I did an analysis of that through a tool called PISA to count how many connection points, there are about 60 connection points. That's a lot
Yeah.
to get right. So maybe the way to illustrate the importance of getting your connections in order, something that's fragile, it's like a house of cards. Things have to be connected. And so one, Dr. Dan kept saying, well, if you're not calculating natural selection, I'm not interested. And it's like, well, you get one shot at it. Otherwise, it's either dead or it's not going to be selected if somehow the cell figured out how to live without a topoisomerase. So you can, and there's something I need to show, and evolutionary biologists, I don't think appreciate this fitness landscape. And this is, we really know this in evolutionary algorithms, but I think it should be brutally obvious also in biological cases. So, okay, I think this is in computation. Okay, so see if I could show this. The diagram on the right there is a fitness landscape. And the way this works is kind of like anti-gravity, conceptually. If, for example, you wanted to have something that's really fit, and you're kind of stuck in, you started inside this mountain of selection pressure, the selection pressure is going to optimize you to that point. If you wanted to change your architecture to get something better, like here, you actually have to resist selection pressure, somehow get over here, you're close enough, you've changed enough amino acids, and then it can start to pull you and evolve you. So the problem is, what this shows is natural selection actually prevents change. Natural selection, you're going to break something first before you turn it into something usable.
Yeah.
And I'm just like, guys, do you not see that? Why do you think selection's going to work? Because they keep being deceived by all this morphological stuff and really kind of cherry-picked examples. What they need to have is showing that it's the fitness landscape, natural selection, or I don't even want to call it natural selection. Differential reproductive abundance is a mechanism that is going to prevent evolution of complexity, because you have all these valleys. It's preventing what they called Darwinian selection. It's actually going to prevent evolution of a new form, because it's to go from one spot to another, it's just not going to work. I think you get that. I don't know why most evolutionary biologists don't understand it. Social contagion. That's the best explanation.
I mean, social contagion, and the fact that if they ever said anything like that, grant money would dry up like the Red Sea in front of Moses.
Okay, so how did this involve ploidy? Why did I think of you? Michael Lynch gave a very compelling problem. He said, what if you're trying to transition? Let's say hypothetically, this is monomeric to begin with, which is kind of, for potassium ion or topoisomerase, that's ridiculous. But let's say hypothetically, so you have now a new allele. So you have one allele that goes monomeric and the allele that's for dimeric. he said there's going to be a complication there because one allele. How does this work in Mendelian? See, this is why I think it's good you and I talk about this.
So if you have one allele that is for monomeric and the other for dimeric, and you're in a diploid system, you are going to have one of those two alleles is going to be more dominant over the other, according to classic Mendelian genetics. And whichever one is dominant is the one that's going to be expressed when it's present. Now, there's codominance and incomplete dominance and other things that can affect that.
Oh, okay. Thank you. Thank you. I just realized all dominance is actually incomplete. It's just so the incompleteness is just so tiny that it's not perceptible. The reason I actually looked at that, and it's only like in the last 10 years they began to figure this out. So like the expression level of a dominant allele is like 100 times more because they started to study how the circuits work, how the regulatory circuits
which will make pink flowers, but if you're making proteins is a problem because it could means that there's going to be less proteins of the correct form than there should be.
Right. Correct. So this is interesting then if you have a, would this be heterozygous?
Oh yeah, that would be heterozygous. Having one of each would be heterozygous. Yeah.
All right. So he was pointing out, okay, so let's say one is the dimeric form, one is the monomeric form. And he said there's going to be complications there.
Yeah.
He said, how would that be selected for? So that's why I thought of you when I was reading that. I'm like,
how would it be selected for to have both of those at the same time? Is that what he's asking?
Well, why don't I read what he said?
Yeah. Why don't? Well, where is it in the paper? And I'll read it and try and make sense of it because I've got the paper pulled up. But just where is it in the paper?
Okay. He did find something interesting. Most of these, you have very rare odd numbers of multimerism. It's usually even. Called it even meric or odd meric. So let me see. I think.
Is this on the rarity of trimers, Sal? Is that the section?
I don't know. I only read through half the paper, and I was just having such a good time, but I was getting tired.
Right.
So,
So while you're looking for this, can I ask a question? Because I don't know the answer to this. In the case of that you're talking about, where there's one allele that has a dimer structure and one has a monomer structure, do those two combined into a trimer, or do those two circulate as independent monomer and dimer structures?
in general, it's going to be even number. If you have a dimer, I don't think I even found any examples where you had something that was ever in a dimer form becoming a trimer.
Okay.
They're rare. Put it this way, rather than putting myself at risk, I'd say it's rare. But I found the relevant passage, and it's thick. Okay. But this is what caused me to give you a ring. It said, in diploid species, some dimerizing mutations may have a deleterious effect, may have deleterious effects in heterozygous individuals as a consequence of heterotypic complexation, a process that can greatly reduce the probability of establishment of the dimerizing allele. So, do you remember that nasty equation we were working with that one Saturday night?
Vaguely, yes. I tried to block that out of my brain, but
Okay. So sickle cell anemia is heterozygous advantage.
yes.
He, I think, is referring to the inverse situation, heterozygous disadvantage.
Okay, so this would be where the homozygote is favored, which was, I'm remembering correctly, is under dominance.
Yeah, this is now he at least gave a reference. I mean, if we really wanted to torture ourselves, we could look at that. But this is what kind of stimulated my interest here.
He referenced himself.
Oh, so let me just go a little earlier. Numerous human disorders are known to result from inappropriate protein aggregation, like no duh.
No, right? Yeah.
And highly expressed proteins are especially vulnerable to promiscuous interactions. Third, in diploid species, some dimerizing mutations may have deleterious effects in heterozygous individuals as a consequence of heterotypic complexation, a process that can greatly reduce the probability of establishment of the dimerizing allele. So this is where he's actually, this is where I say he's relatively sharp in his math.
Mm-hmm.
He says his math skills aren't that good, but relative to most evolutionary biologists, he's
relative to most people, his math skills are pretty good.
Yes. So, I'm sure the math is there, but he was saying.
So I think if I'm reading the statement that you read to me correctly, what he's saying is that in a diploid species, if you go from a monomer to a dimer, so you're creating that's why it's said a dimerizing mutation. You're taking something that was not a dimer before and is now making it a dimer. You may have deleterious effects in heterozygous individuals. So the reason for this is previously you didn't have a dimer, and whatever you had before, whether that was a monomer or a octamer, whatever, you don't have that anymore. So you now have two different structures of the same protein floating around. And of course, this depends on dominance, but you have two different structures of the same protein floating around, one of which may or may not work.
Yep.
And because of that, it likely is going to have a negative fitness effect. Because the body's spending energy creating something that doesn't work. And what that's going to do then is reduce the overall fitness of the organism. Now, it may not reduce it extensively. It depends on how much of an effect it's having, but it will reduce the overall fitness of an organism. And if the overall fitness of an organism is reduced, that means its likelihood of producing offspring is reduced. And therefore, the likelihood of that allele being established in the population is also reduced.
Right. He's basically, again, that fitness landscape.
Right. Yeah.
By the way, let me acknowledge Mark O'Connell. Are you still there, Mark? I'd like to thank all the veterans of the United States military for their service to the country,
yes.
Sir.
Yes.
God bless you. I salute you and thank you.
He asked a question right above that, Sal. I don't know if it's something you're more qualified than me.
Who knows by what mechanism does topoisomerase cut? It is mechanical process like scissors or some molecular unbending process, rebounding process, or combination of both. I actually don't know. That's a Joe DeWeese question. Yes, thank you again, Mark. And I couldn't help myself. It was really nice to hear Zelensky just salute our troops. He is a politician, but he has a country that has a very strong Christian conscience relative to the USA.
I would not say that, Sal.
Okay, maybe then I'm mistaken. All right. Anyway, reason I said that, a lot of his messages, even though he's Jewish, like he had an Easter message, it was beautiful. So I just take it as that he's an actor. He did a good job. Okay. So anyway, I'm grateful that for the salute to the American veterans. That was a beautiful tribute to them. So, and then thanks to all the veterans, including Mark O'Connell. So going back to this, and I'm sorry I couldn't answer your question about the topoisomerase there, Mark. Thank you for joining us. So, and he's talking about the difficulty of going from one to another. So that's what this paper was about, because I was talking about evolution of multimeric forms. I'm glad I stumbled on this because I don't want to make a blanket statement that all multimeric proteins are difficult to evolve. I would have made myself vulnerable. So one of the benefits of me interacting, occasionally debating topics, is it forces me to go back and double check my own claims, because there is a lot of literature out there. And so now I feel better that I did double check, and I have to temper some of my comments. I'm not going to make a blanket statement, multimeric proteins are difficult to evolve. Certain selected ones are definitely going to be problematic. So, and that's where the ploidy stuff came in. But I began to have some other thoughts about paralogs. And let me see if I can bring up that diagram of paralogs. And then we can actually get to the topic of the night that I advertised, but then I started to have second thoughts.
Only took us an hour and a half. We're not doing too bad.
Yeah, don't do that. Oh, here. Okay. Let me mute my mic a little bit. Siren. Oh, so I keep hearing the word ortholog. I kind of know a homolog. Ortholog. Ortholog. I use ortholog so much I keep forgetting. I mean, I use the word homolog so much, and I know paralog. So let's see the definition of a ortholog. Is any of two or more homologous gene sequences found in different species related by linear descent. Any of two or more homologous gene sequences.
So theoretically, any homologous gene is an ortholog, according to the evolutionary model.
If it's in different species.
Correct.
A paralog is in the same. Okay. Any of two. So if it's a ortholog, it's automatically a homolog. Any of two or more.
Yes, that would be true.
Any of two or more homologous. Okay. Any of two or more. Okay, so we could say like all the orthologs of topoisomerase alpha. Okay. And this is where it's funny because, all right, so we have like this is actually a nice diagram because it raises the issues. And maybe I was thinking this is more of a problem for evolution than it is, but it's still an interesting question. So we have a topoisomerase alpha and a topoisomerase beta in humans. So those are paralogs within our genome.
Correct, yes.
And they're homologous to each other. But what's interesting is you would say the topoisomerase alpha, there are orthologs the alpha ortholog exists in frogs, human, and mice.
Correct.
And
So
beta-
according to the model, all well, at least those humans, frogs, and mice all got their alpha topoisomerase gene from a common ancestor. That's what makes them an ortholog.
Right, and it would also have to get the beta. And I was just thinking about this.
Correct, but not necessarily from the same common ancestor.
This is kind of what is. I had to really wrap my head around this. This is one of those where we'd have to really think and have the data plots. But one of the problems is, as I was thinking about this, because they have all these molecular clocks and theories and stuff. If like say the alpha homologs are very similar, and then the beta homologs are very similar.
Orthologs.
Okay, so the alpha orthologs, and then the beta orthologs across species
are very similar to each other.
Right. But yet, I was trying to think how this would be a problem for evolution. It may or may not be. My math instincts were kicking in, but I was kind of sleepy at the time.
Never do math while you're sleepy. That's always a bad idea.
So that's what I wanted to ask you about, but now that was premature.
Let me chew on this diagram for a second and see what I can come up with. So, I think we'll start of the problem will be the gene duplication is assumed at the outset. We can duplicate genes, but you'd have to create new functions for those genes unless you're going to argue that the alpha and beta topoisomerase gene, both functions existed prior to the gene duplication and then it was subfunctionalized, which I suppose you could argue that if you wanted to. But you don't necessarily, that wouldn't necessarily, it still doesn't explain where the original function came from, which is what this is trying to explain. Now, in terms of orthologs being different in having alpha orthologs and beta orthologs and then being homologous, this is highly phylogenetic dependent. And phylogenies are unreliable, particularly gene trees. And evolutionists know this. Gene trees conflict with each other all the time. This is not unheard of for gene trees to conflict with each other. And if Joel Juff's still here, I'm sure he'll back that up. Gene trees within the same set of groups of organisms will conflict with each other. And I've been doing some playing around with coelacanth genes recently. And I mean, I've gotten all kinds of gene trees. I've only done probably a dozen gene trees, but I've gotten all kinds of things. I've gotten where they were monophyletic, paraphyletic. I've gotten where they were most closely related to caecilians, which are legless amphibians. I've gotten where they were most closely related to non-bony fishes, chondrichthyes. I've gotten where they're most closely related to bony fishes. I've gotten where they're most closely related to watsons, which are a bird. So gene trees are something of a flaming dumpster fire. You never know quite what you're getting out of them. And so to argue from the gene trees of the alpha and beta topoisomerase genes that you can trace that back to a common ancestor, I don't think that's a fair assessment, because you can get any gene tree will tell you all kinds of different things, and it of course depends on what organisms you include on the tree. But I'd be very hesitant to use a gene tree to say anything at all about the ancestral patterns of organisms, just because gene trees are known to be highly conflicting with each other, which would be a problem in particular if you had a gene tree for something other than topoisomerase. I don't know, maybe you use it for the proton pumps or the potassium ion channels, and if you did a gene tree for those and they used the same set of organisms but came up with a different tree, you'd then have some interesting questions to ask, because which tree is correct? And of course, evolutionists will tell you, and you read their phylogenetic literature, they'll tell you that probably every phylogenetic tree is incorrect. But it's an approximation. It's a hypotheses about the past, which, okay, if you want to make those hypotheses, okay, fine, we can make those. But then don't turn back around and tell me that you can demonstrate that this gene duplication happened this many years ago based on gene trees. Gene trees are not reliable, and everybody in the literature knows that. You can maybe it's nice to trace them back, maybe, and maybe we'll figure out where that duplication supposedly happened, but they conflict with each other. And I'd be pretty willing to bet that this simplified gene tree that you have here, if you actually did a gene tree of all the orthologs of the alpha topoisomerase gene and the beta topoisomerase gene, you'd get two wildly different trees.
Yeah, I feel bad now that like I said, I was doing the math, so this is all kind of premature. But hey, it gave me a chance to hang out with you.
Yeah.
There were. Oh, I'm sorry about that. Sorry about that. Didn't mean to show that. So let me go back to that paralog thing. I was just as you were talking. I was also. I'll give you 2 data points. I posted something on hostile pseudoscience. Swami Das.
Yeah. I don't know why you waste your time there.
Well, no, because I wrote some really good stuff there. It's part of the editorial process. And I have to pay $1 a month to get back into the forum.
Seriously, he's charging for his forum now?
I don't know. I want to get back into the Peaceful Science forum because, it's the Patreon. It put me in the Patreon page. I used to be able to. Okay, I'm going to visit the forum, see if it'll let me. Discourse Peaceful Science.
It looks like that's still free, which, as far as I can tell, anyway.
Okay.
Although it doesn't look terribly active.
Sorry?
It does not look terribly active.
Well, because I'm gone.
Yeah.
I think they really wanted to just have a feeding frenzy over me.
Yeah. But just to be fair, I mean, the vast majority of the topics that I'm seeing on here are closed and haven't had any activity for weeks. There's only I think 2, 3, 4, 5, 6. There are 6 open topics on that forum right now, and one of them is basically a feeding frenzy about the midterms, which wouldn't be there any other time of the year. So it looks like they average about 5 open topics at any time, which isn't very much.
All right. I gotta just laugh here. Mm-hmm. Of course, they're just going to be. They're usually mean. I mean, they've. It says in Proverbs, correct a wise man and he'll love you. Mm-hmm. Correct a fool and he'll hate you. over the years, Arthur Hunt he started out being nice. He started to progressively get nastier and nastier. The more that the more education I got under my belt, he started to go ballistic.
Mm.
Now he's carrying, saying all sorts, just all these kind of like really nasty things about me, saying, you're so pathetic. You can't, I mean, it's just, and now that I look at this, it's like the only reason I endured this is I was trying to get editorial comment, but it, when it gets time to when it gets to the point, it is that bad. I just want to quit. So I did find what I wanted to share with you because this is multimeric. So I'm going to show you something. So first the diagram here. So let me bring it up. And I'm glad I kept this because, all right, so we have the microtubules.
Mm-hmm.
And you may have heard that.
Yeah, I know what microtubules are. Yeah.
It turns out you need the paralogs, beta, alpha, beta, alpha. And I said, how can you have the system work? See, it has to walk. It looks like it takes steps on all the betas. The alpha is a spacer.
Whoa. Hang on a minute. You're telling me that for kinesin to walk along the microtubules, it has to have both paralogs present at the same time?
Actually, it's 4 paralogs.
Holy jump.
The gamma starts it, is the root, the alpha, beta alternate. And I saw this right away. I was like, there goes gene duplication. You can't just...
Yeah. That just gene duplication just went out the window.
You got it.
That's impossible. That is bloody impossible. You can't do that. It's not. You can't have something as fundamental to a cell as kinesin, which I think is like almost ubiquitous across eukaryotes. I don't know about prokaryotes, but eukaryotes, it's very common. You can't have something ubiquitous like kinesin be dependent, which requires now not 1 but 4 paralogs, which supposedly are derived from gene duplications. No, this doesn't work. It can't work. I'm not saying gene duplication itself can't work, but it can't work in kinesin.
Exactly.
The kinesin cannot be a product of gene duplication. It's impossible.
So what we're looking at is the alpha and beta paralogs are tubulin. And then the kinesin walks on the tubulin paralog, the beta. So this is the kinesin here, and this is this. But the problem doesn't go away. I mean, and let me see.
Oh my.
See, dimers of tubulin. So it uses the word, but this isn't just a dimer. This is like I don't know what you'd call that. A 1000-mer. I mean, it's a dimer, but then you have many of these pairs.
It's stacks of dimers. It's like a, I don't know what you call a
100 dimers. Yeah. And so I threw this up there, and of course he said, "Oh, you haven't looked at the paper I posted," and I was being attacked so fast I didn't have a chance to read everything. Mm-hmm. And he was this Evo grad student, and because he's an evolutionary biology student, I'm sorry to be nasty, but don't have a very high opinion. At the start, if he tells me he's studying evolutionary biology, I said, well, there goes someone's lack of critical
chronology. Okay, good to know.
Yeah. Oh, here it is. Here are the paralogs. Alpha, beta, and gamma. So you have gamma tubulin, and then you have alpha, beta. So gammalin. So these, you need all 3 simultaneously.
So in nucleation and polar orientation of the microtubules, we'll be jumping.
All right. So Evo grad student, just because he found a speculation, he said it's solved. And oh, here's another picture of it, because I saw this from Bruce Albert's Essential Cell Biology. So you can see how this lines up. There, it's even more complicated. So you have kind of the base one here, but it's actually, you see how it circulates like that?
Yeah, it's
Isn't that
circles.
cool? Yeah. Okay, so that's a tubulin dimer. And I'm sure there's a biophysical reason for that.
I'm sure
Okay.
there is.
And so I was making fun of Evo grad student.
Yeah, I mean, Gene, I had already. So the first graduate school class I took was a genetics class at of course, the graduate level. It's an evolutionary class, and it's they told me that gene duplication was a regular thing. Now I was skeptical of gene duplication when they told me about it, but I didn't have hard and fast evidence for why. There was something that it didn't feel right to me, but I wasn't quite sure what it was. Now I know what it was. That doesn't work. Now, I'm not saying that gene duplication could never happen, and I think it, at least on a polyploid level, you could have it. I think that's considered, although I've got my own questions about whether polyploidy is a thing, because as it turns out, polyploidy is also evolutionarily defined, because there's no set how you, for plants, they determine whether something is polyploid or not based on whatever a set standard is for the genus. So it's completely arbitrary. And it might be that, now, again, I'd have to dig into this more. I'm still working on this, but it could be that the label polyploid is being given to organisms that are not polyploid. Instead, what you have is you have the set for the standard for the genus, X equals the haploid state is 7 or whatever, is actually represents chromosomal loss rather than chromosomal gain.
Okay.
Now you can induce polyploidy. Polyploidy is a phenomenon that does exist. You can induce it in the lab, so it is a real thing. It just could be some of the they talk about like 40% or 60% of plants or something like that have undergone polyploidy at some point. Like, that number is undoubtedly inflated, because it's a lot of genera probably have a less polyploids than we're told they have.
Wow. Okay. As you were talking, I was just scrolling through this. I got to tell you, now that I look at this, I don't know how I was able to deal with these clowns. And I'm sorry to be harsh, but they're clowns.
So if it makes you feel any better, Sal, that was how I knew who you were when you showed up on SFT. Is I followed that forum. I didn't post, but I followed it, and I would see you dealing with these people and I'd go, man, he got more patience than I do. I'd just called him an idiot and been done with it.
Well, the thing is that they may be idiots, but you got to deal with their arguments, because
right. Although
the
some
argument,
of them, I didn't see very much argumentation, to be perfectly fair.
Yeah, well, that was one thing, is when they weren't arguing with me, I knew I had a good case.
Mm-hmm.
Because I posted something on double-stranded break repair, the evolution of that.
Mm-hmm.
That didn't get many hits. I said, okay, you told me what I needed to know. Hammer that. If I talked about tiktaalik, it'd be endless. Mm-hmm. But I was just probing. They call that in the military parlance, reconnaissance, move forward, draw fire, or reconnaissance and force. Yeah. Just trying to probe where their positions are and where the weaknesses. But I had, now looking back, I said these guys were just. I think all the ad hominems and all the nasty comments is telling me, actually, the more that either they're dead silent or the more that they poured it on, I said, I have a good argument here. because their responses had no substance. But I said, every now and then someone will make a good comment, but there's some really, there are characters there that I wish I could block and not see. John Harshman's one of them.
Yeah.
uh,
Matheson's another one.
Who?
Matheson's another one, if I'm remembering correctly.
Oh my goodness! And you know who he is? He's
I know
the
exactly who he is. That's why.
editor-in-chief of Cell Reports.
And if I recall correctly, and I'm going to verify this before I state it, but if I recall correctly, if this is who I think it is, and
It is. As in a certain event at Calvin College.
Yes, that's what I was thinking.
Oh my goodness! You know, he was just taking, just insulting the leadership. And then when he got in his indiscretion, the leadership made him pay. They wrote a letter to the entire student body saying, we do not tolerate certain behaviors from our faculty. This is what Steve Matheson got caught in. Wrote a letter to every student and faculty member about Matheson.
He's a really bad actor.
And then they got it on Fox News in the local television. So they let him know that they didn't, they're going to take a lot of revenge on him. That was vengeful, and I actually kind of enjoyed it myself. And I said, so you've been at a Christian college and you're doing this, and now you get caught, with an undergrad.
Yeah.
And you've been representing yourself as a Christian, and now you're an atheist, and you come out and say, yeah, I'm so happy. Okay.
Yeah, and as it turns out, our buddy, the guy who runs Unpeaceful Pseudoscience, let him write for him. There's an article by Matheson on the Peaceful Science website.
Who wrote for who?
Matheson wrote an article for the Peaceful Science website, the actual website, not the forum, the actual website.
Okay. And the irony is when the owner was trying to curry favor with my boss, he recruited Matheson to review my article, and of course they were trashing it. But he said your conclusions were right, but it's not.
Yes, I remember that.
And they actually gave me a data point, and I since corrected it, sent it off to peer review. That version. He said, well, your conclusions are right, good paper, but it's out of our scope or it's too late. I'm just like, yeah, 7th rejection. Most of the editors said we're correct, but we're not going to publish. I got kind of fed up with the whole process. So I'm now looking in the Middle East journals to publish. John said, you're not going to publish in a creationist journal. You're going to publish in a secular journal. I don't care if it's in the Middle East. Or India.
Probably a better chance over there because at least some of them believe in some form of deity, so they actually let you publish it.
Yeah. So he said, John, you're really determined not to have me publish in a creationist journal. He said, yeah, I don't want you doing that. We're going to put our foothold in the secular world. So that's on the back burner, because a lot's happened to me, as you know. So that was epic.
Yeah, I remember that though. I remember that. I remember that. I remember that incident loud and clear when you got that paperback and you said, "Yeah, your conclusions are essentially correct, but we're not going to publish it." I remember that. I did not realize that was Matheson that had done that though. That adds another level of petty to that individual. Well,
no, I didn't submit to his journal.
Okay.
But he told me he wouldn't. He's not going to help me. He wouldn't, and so that's so, I appreciated.
Give him another undergraduate to have a fling with. He'll do it.
Oh, that was me. Okay, so going back to the, I'm sorry.
I'm sorry. That was probably uncalled for, but it's after midnight, and I have about as much respect for that individual as I have for I won't go there. But yeah, you figure that out on your own.
So this all triggered. See, now it's become part of the language. The word being triggered. This all got started because I was looking at the I was looking at peaceful science because we were looking at this tubulin paralogs that make microtubules, and so and there was a guy named Mercer. He was also another nasty individual. But then I began to read what what's his name, Evograd wrote, and my jaw dropped. I started to laugh. It was so bad. So if I can find it, I'm gonna look. I need, because it was too funny. I got to read you what he thought was a great explanation. Okay, so Harshman's just another joke.
Isn't Harshman the one that identifies as an avian phylogeneticist?
Yeah.
No, phylogenetics is a joke, so that explains that.
And let me see. I hope you'll forbear with me, because I think it'll be worth getting the laugh out of this.
Fair enough.
And that otherwise I wouldn't be wasted. I wouldn't want to make you wait for anything, but this was just so funny. So now you know the problem with that, and unfortunately I can't just block all these guys. All right, let's see. Okay, so he solves this problem with speculation. I mean, Evo Grad thinks this is a good explanation, and he was highlighting it and posting it just for my benefit, and I just laughed. I really encourage people to read this paper. It's extremely comprehensive. Tubulin, the protein subunit of microtubules, is an alpha-beta homodimer. In this chapter, a hypothesis on the evolution of the tubulin molecule is proposed, based in part on reports on the structures and functions of different forms of tubulin and its relatives. The concentration is on three main areas: evolution of the vertebrate beta tubulin isotypes. In addition to providing a clear idea about the relationships among these isotypes, recent data suggests that tubulins may have functions that do not involve being in a microtubule, namely that it can function as an isolated alpha-beta dimer or as a non-microtubule polymer. Examination of both the entire tubulin superfamily, which includes not only tubulins, alpha, beta, gamma, delta, epsilon, eta, and others, but also a variety of prokaryotic proteins. The hypothesis is presented that the common ancestor of all these proteins formed a filamentous curving polymer that used energy of GTP hydrolysis to apply force to nucleic acids and or membranes, that this common ancestor may have been co-eval with the first cells. A variety of chaperones, motors, and MT-associated proteins may have co-evolved with tubulin, and their histories illuminate that of the tubulin. The branched, highly negative charge C-terminal domain presents an alpha and beta tubulin appears to be relatively recent addition to tubulin. These hypotheses is presented that the C-terminal domain may have been a prebiotic origin.
This is their answer.
This is their answer. And then it gradually developed into a protein serving particular metabolic functions whose genes eventually became fused with those of alpha and beta tubulin. So we have this precursor that is prebiotic. Somehow you had alpha and tubulin paralogs, okay? And it just happened to connect to the right gene locations.
This is what the highly vaunted Evograd is selling as the answer to this.
Yes, he's saying, I encourage you to read this. And like I said, this is an example where evolutionary biology and critical thinking don't mix. How the heck are you going to get this thing of prebiotic origin that ends up using? And you know what's really bad? How do you know it's prebiotic origin? Do you have it? How do you prove that?
Yeah.
You happen to have the same similar tail on both, and you're going to say, oh, that had to be a prebiotic origin. So you had a gene duplication that made alpha and beta, and then we're going to simultaneously attach two pieces to the ends.
No.
Could I make a cartoon out of this? I wish I could. If I were
this reads like Abbott and Costello. I mean, this is insanity.
insane.
This is insanity. And Evograd thinks this is a
good
viable explanation. I mean, I remember when certain people, including certain people who were in this chat not that long ago, were pumping up Evograd as the ultimate refutation of Jeanson's book. And I remember reading some of it and going, it wasn't that good to start with. But now I'm like, if he thinks that's a reasonable explanation, why should I trust anything this guy says?
Oh, the stuff with zinc fingers. See, maybe this is why I tolerated it, because you get gems of stupidity like that. First order
published in
stupidity. I'm sorry.
I said I can't believe that gibberish was published in a peer-reviewed journal, let alone that somebody else thought it was a good idea.
You see, this is why I enjoy hanging out with you. You're the one person that would realize this was a joke and you would laugh. Anyone else would think this is so sophisticated. Even an evolutionary biologist. That's the funniest thing, is he thinks this is a good explanation. What's worse is all his compadres there, from John Harshman to Swami Das to Steve Matheson's, letting him get away with it. You know, Art Hunt. I'm just like, you're going to let this garbage go, guys? You know, come on.
It's just garbage. You cannot sit here and tell me that something could somehow create itself prebiotically and then later on be incorporated into postbiotic genes.
The paralogs, nowhere else, I guess.
And it just happened to be incorporated into two paralogs after their gene duplication. It didn't incorporate anywhere else in the genome. It just happened to pick those two paralogs. What the? Do you explain? How high were you when you wrote that? Yeah.
Okay, my neighbor, he has this posted up in his on his wall in Latin: non potes hoc stercor sursum, which means you can't make this stuff up. Okay, I'll use the polite word.
Yeah. I mean, this still doesn't quite match aliens sent the octopus to Earth and we all evolved from the octopus, but it's up there. Oh my!
It's up there.
Oh, that's
okay.
painful.
So the hypothesis is presented that the C-terminal domain may have been of prebiotic origin, and that it gradually developed into a protein serving particular metabolic functions, whose gene eventually became fused with those of alpha and beta tubulin, Finally, some experiments are proposed that could illuminate the probability of these hypotheses.
Gene fusion?
Okay, so
even if this thing could have somehow got itself into a gene postbiotically, which is garbage, but let's just suppose it did, it would have to somehow fuse with alpha and beta tubulin genes, which, as far as I know, there's no process for that. Maybe you could argue that it's a jumping gene, it's a movable element, transposable element. But even so, even if you nestled it in right next to the alpha and beta tubulin genes, there's something called linkage disequilibrium that decays over time because of recombination. It wouldn't stay there. Like, you'd have to protect it by a chromosomal inversion.
Oh, can you run that by me again? That was really that actually was sophisticated.
Okay. So even if we grant his original premise that we somehow get this gene from prebiotic goop, let's just say that we get this gene. It would have to somehow end up on the end of both the alpha and beta tubulin genes, and it would have
right.
Now to get there, it would almost have to be a transposable element. Now, most transposable elements cause mutations, so we've got problems right there to start with, and bad mutations. I mean, like deleterious ones. So we got problems there to start with. Well, let's just give them that too. Let's give them that too. There's something called recombination that happens in eukaryotic genomes. And what recombination tends to do is it tends to break up associated genes. So if you have a group of genes that hang out together, over time, recombination decays that. When they hang out together, it's called linkage disequilibrium. Recombination breaks that down over time. It tends to decay towards zero, which is linkage equilibrium, where there's no connection between the two. So the only way that I know of to protect that from recombination is to either somehow fuse it onto the gene, so it's actually the same gene, which you would have to do on both alleles at the same time. And on both genes at the same time, which is just insanity right there. We can start with that. And as far as I know, there's no process for this. We can add that to the fire. But to make things worse, as far as I know, there's only one way to protect against recombination, and that's to do a chromosomal inversion, which is rare and usually negative, where you basically have a chunk of the chromosome that flips around. So it instead of facing its normal way, it will flip. It will do a 180-degree turn so that the end will move into the middle of the chromosome, and the middle will move to the end. And maybe there's another way to do this. Maybe I'm missing something. That's conceivable. But chromosomal inversions are bad in general. Occasionally they're not, but generally they're very bad. They're very, very bad. And you'd have to get and I don't know if the alpha and beta tubulin genes are on the same chromosome.
How would you protect it? So what was the purpose of the inversion again?
So the inversion prevents recombination because you no longer have the two genes are no longer opposite each other on the chromosome.
Holy smoke. So recombination can make one of them go away.
Well, recombination can effectively move one of them away. It can move away from it on the chromosome because of the crossing over.
Yeah. Right. Wow. I found my response to him, and I wish I had. That's the problem. Is in text you can't put how much you're smiling and laughing at these guys. So he said the hypothesis is presented as the C-terminal domain may have been a prebiotic origin, and it gradually developed into a protein serving particular metabolical functions, which are unproven, whose gene eventually became fused with the alpha and beta tubulin. And I said, this would be a rather fortuitous event, not much different than a miracle. Thanks for the paper. Like I said, common descent needs miracles to make it work. So that was the polite version. And it says a variety of chaperones, motors, and MT-associated proteins may have co-evolved with tubulin, and their histories only illuminate that of tubulin. Same for this coevolution. Did the Diane microtubule binding domain co-evolve too to connect the newly evolved alpha and beta? Okay, so let me just, for the reader's benefit, show what I mean with the problem of coevolution. And I have a slide from Bruce Albert's Essential Cell Biology, which is where I learned about microtubules and tubulins and paralogs. Mm-hmm. And when I was reading this, my jaw was dropping. I said, "This is, this is gold." If we look at it this way, okay. So, what would coevolution? Okay, so just think about that. Okay, that picture of the. Let me see if I can find it now. Oh, there it is. Okay, this nice little picture here. So this, you have for this to work, you have to have a connection of the kinesin's foot touching the beta. I just realized something. I wonder if this is a monomer or a dimer. In either case, it's complicated because you have to have, these have to be identical feet, I think. I don't know.
Looks like it might be a dimer, Sal, based on the way it's drawn there. It might be a dimer.
Okay, but in any case, it has to be, the first thing is it has to be the right length, this arch. Mm-hmm. The next thing is it has to connect, it has to recognize beta. Because this thing's floating around, it's going to land here, and it has to know that it has the foot on the right spot. Right, the shoe, the shoe fits, so to speak. Yep. And that's a binding domain. So now let me show this diagram of what's involved in that. You have to have complementary shapes. So Bruce Alberts is illustrating this. This is Essential Cell Biology. Not only do you have the complementary shapes, you need complementary charge distributions. This is the other thing. The third requirement is it has to be the right adhesiveness. The correct term is binding affinity. Even Lynch will use the word adhesiveness. So like I used to have rolls of like duct tape. So you could think of duct tape being kind of strong, painter's tape, masking tape a little bit less, painter's tape even less, and then Post-it tape just really, really weak adhesive. There are situations if you can imagine that you do not want kinesin grabbing on too hard.
Mm-hmm. Yeah. Just like you wouldn't use duct tape to put on the frames or around of your doors when you're painting. It'd be too hard to go off and would leave a residue.
Right. So you don't want the Velcro sticking too hard. So it's three things. It has to have the right shape, the right charge distribution, and it has to be the charge. The presentation of the charges has to keep it from being too strong or too weak. It has to be strong enough. So you think about this, that kinesins, I guess if it's hauling anything, it has to have to be attached strong enough so it doesn't get pulled off, but it has to be weak enough that it can walk. So it's just like your shoes. You want to have some grip, but you don't want it to be too slippery. And to engineer this is not trivial. So what does that lead to where he was saying this has to co-evolve? I'm like, do you guys not have any critical thinking? And we know the answer to that. So what would have to happen is, let's say the left side's beta and the other side's the kinesin, or vice versa. If you evolve this, you're going to have to evolve the other one simultaneously.
Okay. Okay. Yeah. Nope.
I mean, they could experiment. Why don't we knock out pieces of this and see if it lives long enough to evolve the other part? So let's compromise the beta.
I got news for you. You knock out either one of those, you got a dead cell.
You got a dead cell. And so Evograd was like, "This is such a great explanation. I encourage you to read it." Yeah, I say I sooner believe in miracles because what you describe is
Nathaniel Jeanson. Like this is what they throw up there as this guy knows what he's talking about. He read Jeanson's book and he can diffuse all the things that he can't. What it? I'm sorry that was my insanity meter just like filling up to maximum. Like what do you? This is their answer. This is their best warrior? Like, this is the guy they're sending out for they may as well send a 5-year-old out. And by the way, I'm not saying he's dumb. I'm sure he's smarter than I am. That's not hard, but I'm sure he's a heck of a lot smarter than I am. But he buys that? That's his, everyone should read this. This is a great idea. Sheesh.
And I thought young Earth creationism was outrageous.
Yeah, good. No, I mean, I've seen some outrageous stuff get tossed around occasionally, mostly by people more of a layman level in creation. I've seen some ridiculous and outrageous stuff, but that takes the cake. Like,
these are the gems from peaceful science.
Oh, good heavens!
They're not many. Okay, there are nuggets of gold in all that poop. So that was a good one. All right. So before we close, I did need to tell you because you mentioned coelacanths.
Mm-hmm.
I didn't even know that's how it's pronounced. I always thought it was coelacanths or something. Or anyway. Why don't they spell it with an S and we wouldn't have problems. Anyway, I think you might get a kick out of this. Let's see, tetra. See if I can get a diagram. So I heard the number somewhere. The tetrapods diversified into 20,000 species.
Okay.
Some gigantic number. And let me see. Let me see if I can grab this. So the
Sarcopterygian fishes.
Yeah, Sarcopterygian fishes. So, and I was getting. Oh, this is so funny. All right, I'm gonna. I found a clocking problem. In addition to the problems you found. Okay, so let me bring this up, and maybe we actually. I hope this was this was fun for me.
Oh, I had fun. I lost my mind, but I had fun.
Okay, this is the nerd version of Saturday Night Live.
We're funnier.
Yeah.
They just lecture you.
I'm sorry, what?
I said we're funnier. We actually make jokes. They just lecture you.
Yeah.
So of course, the evolutionists make our jokes for us, so we don't even have to even write.
I know. We got great writers. You just need to
just go read the peer-reviewed literature long enough. Anyway, go ahead.
So actinistia,
Actinistia. Yep,
coelacanth,
that's right.
and dipnoi, lungfishes, and then tetrapoda.
Tetrapoda,
Okay,
yep.
so this is a living fossil, and so is this.
Mm-hmm.
I did a preliminary study with the COX-1 mitochondrial gene. And the distance of this to this, they supposedly split, what, 300 million years ago or whatever, 100 million?
Well, yeah, I don't know how long ago the split was. The last living coelacanth was supposed to be about 68 million years ago prior to them finding it in the early 1900s.
Okay. The thing is this is very similar to this.
Yeah.
This one, you have 20,000 representatives on this tree. And my spider senses something really smells about that. You're saying that this guy kind of never evolved. This guy never evolved. But then this guy just diversified and evolved into 20,000 species.
Stasis is proof of evolution, just like diversity is proof of evolution.
What?
Sorry, I'm being snarky. Stasis proves evolution, just like diversity proves evolution.
Proves evolution. I like the way R.H. Rady framed it. He said, this is like the German proverb, the bird. If you see the bird sitting on the dung, that means it's going to rain, or it's not going to rain.
Yep, precisely. Heads I win, tails you lose.
Yeah. So, stasis here. But what was really bad is this means when I compare this, I said between the two, the molecular clock is really, really slow. And this clock is really, really fast. That's what I saw in just my preliminary. And it turned out someone at the Smithsonian found the same thing. And he said, we can't explain why the clock rates are so different. I'm just like, well, maybe they didn't evolve from each other like you thought.
Yeah, pretty much.
I mean, it was bad. If you drew out the branches, you want to have the branching kind of come out kind of equidistantly, but instead you have one really long branch like that and two really short ones for the lungfish and coelacanths. I said, guys, this looks totally wrong, because it would suggest the lungfish and coelacanths had a very recent common ancestor. But I began comparing coelacanths to other coelacanths. Oh, and this is where I was very interested. And now it comes back to me about the ploidy and paralogs, etc., and alleles.
Mm-hmm.
Why is there an absence? I mean, you would presume over 500 million years, you'd get a variety of alleles, unless they had a recent bottleneck, like, say, that would make them look kind of like a most recent common ancestor being very recent.
Mm-hmm.
Why is there an absence of alleles?
Yeah, the problem of the missing mutations. A lot of missing mutations of evolution happened. Where'd they all go?
Right. So, it's like, shouldn't, if, let's go back to this diagram. If you're diversifying to all these creatures, shouldn't we see a corresponding increase in alleles? The problem of the missing mutations. Was that original to you? This first time I heard
the phrasing it that way is original with me. But the idea of the problem of the missing mutations is not. I got it from Jeanson. That's where I first learned it, way back in like 2016. But he didn't phrase it that way. He said, I think, something like a lack of diversity or something like that. But I actually wrote an article on this a long time ago where I think it was titled the problem of the missing mutations on my website. And I think that may have actually been the first time I interacted with Joel Duff too, because he came to rip it. But back then it may not have been completely accurate because I didn't have the background that I do now. That was 5 or 6 years ago at this point. But the genesis of the problem, the gist of the problem remains. There's a huge lack of diversity. We'll use the coelacanths. 50% of the coelacanth genome is repetitive elements. Where's the diversity?
Oh, good one. We don't even need to go to the genic regions.
No, 50% is repetitive elements. Where is the diversity? This thing's been around since the dinosaurs, according to the evolutionary model. Where's the diversity?
And you see, what they'll say is there's a bottleneck. But then what I started doing is I looked at all this. I just randomly went around and see the age of species. Mm-hmm. I said they all look kind of young. Yeah. I wonder why. Yeah,
they all of them, even the most diverse species, look young. There are mutations that are missing. That should be there if evolution were true.
You know what? This is the first time I mean, maybe I've sensed this, but it's nice to hear you articulate it so well. And you cite the examples. This is a nice argument. By the way, is Dr. Duff able to give you any?
This was so long ago that I don't remember. I'd have to go back to the comments of the article. And I may be misremembering. That may not have been the article. It may have been. I seem to remember him commenting on that article, but I could. Again, this was a long time ago. I don't remember for sure. I do remember getting some negative feedback, though. I don't remember exactly what it was, or for sure. But again, the point I'm trying to make here is evolution. The mutation rates that the evolutionists propose. Now, they're going to use a phylogenetic rate, and they're going to say that there's a constant rate of mutations from however long ago to now, and that based on 68 million years, we have exactly how many mutations we would predict because they're assuming what they're trying to prove. But if you actually measure the measurable rate of species mutations based on a pedigree rate, there are mutations that are just gone. They aren't there that should be there if the evolutionary model was correct. If species were as old as the evolutionists say they are, then there should be mutations that are there that are not there.
This thing about repetitive elements, this is the most adept. This is the most skillful use of it because I was looking at the genic region. It didn't even occur to me. Can I ask a dumb genetics question?
Sure.
And this is sincere. I've always thought recombination deals with homologous genes between chromosomes. What about the the intergenic? Is that the right term?
So the non-coding regions, regions. As far as I can recall, and I could be wrong about this, I'm not an expert on recombination. I'm thinking that recombination is only genetic. I'm thinking that is something that would only be genetic. But that said, that there should be a ton of mutations in these repetitive elements because they're not under selection, supposedly.
Regardless of whether it's under recombination or not, I think it would still be a problem that we have these missing mutations.
Right. Yeah. The recombination doesn't really play into this particular question so much. It does create problems for genetic sections like we were talking about earlier with the chromosomal inversion being required to prevent recombination. But this particular issue, recombination is not so much of a problem. One thing recombination can do, though, is if you have a coding region that suffers a mutation, recombination can recombination can, because you have the diploid or whatever set of chromosomes, you can sort of mask a single mutation to a certain extent if it doesn't make the if the mutation is non-dominant, which most mutations are not.
So okay, I'm just trying to think. I think I'd have to think about this hard, but to get a bottleneck to work to clean this out so that you could say I was an evolutionary biologist and I wanted to get rid of this absence of mutation problem. I'd have to have a bottleneck and it'd have to last a long time to clean out, to get everything.
Yes. So the evolutionists refer to getting rid of deleterious mutations in a population as purging. And yes, Godzilla Freak, that the answer to that is yes. Well, it could, at least it can be construed as evidence for functionality. The Evos like to dismiss that. Dan Grau wrote a paper that really ripped on that. But yes, that could be construed as evidence of functionality. But just be aware that the Evos are not going to like it if you use that. They're gonna call you all kinds of names, and yeah. Let's put it this way: Dan Grauer wrote a paper when ENCODE made that an argument like that, effectively, where that was partially entitled "The Evolution-Free Gospel of ENCODE," and he got it published in a peer-reviewed journal with that title. So you should be aware that they're not going to like you if you make that argument. That said, coming back to where I was going, which I don't remember where that was at this point. Non-coding regions. So you had asked me something, Sal, and I can't remember what it was now. It's 1 o'clock in the morning. I apologize.
Oh, I was talking about how long it had to be bottlenecked.
So a bottleneck to get rid of all those, mutations, you would probably need an extended bottleneck in the evolutionary model. Not necessarily, but you would probably need an extended bottleneck. Now, in a creationist model, there's a short bottleneck which doesn't allow things to fix after post-flood, but it takes a while in a evolutionary model to fix certain traits, and for that you'd need a longer bottleneck.
Yeah, this what you've said is just really compelling. I got to tell you, this is all right. I appreciate what Nathaniel, Rob Carter, and John Sanford were doing, trying to get the 6,500 year for mitochondrial Eve and atom. But for me, if you show all these species are younger than less than 10 million years, that still looks, for me, that's a good enough anti-evolution argument for my purposes. And I have since, and I've worked a little bit collaborated, or Russell Humphreys mentored me in understanding his theories on geomagnetism. Mm-hmm. I mean, when I saw the Wikipedia article saying that, maybe I could show it. A picture's worth 1,000 words.
No, Sal, don't show it, because then if Wikipedia editors see it, they'll change it.
Oh no. Well, it is deep into the peer-reviewed literature at this point, like climate change. It's except it's actually good data.
Oh, so not like climate change, in other words.
Yeah. Let's see. And I asked if this is the same diagram that is in Russell Humphreys' paper, and he said yes. So let me show it to you. And this will relate to why it's good enough, the genetic data. 17 stuff that's before 1700, you can just circular reasoning.
Who knows? Nobody knows, right?
This dark blue line is really carefully. The first time we actually had the instruments that could measure the strength of the magnetic field, all of this is inferred. So,
So
but
just the
you
dark
see
matter
how
stuff.
right? You see, this is going down,
That's the
and
axial dipole strength of the magnetic field.
right. So the axial dipole is like the magnetic field as far as north and south. The variations we can call like these other like multipole pole fields. That's that may be more of a mathematical um, kind of convenience to model it that way. But it's basically north and south with some perturbations, with some uh, things that would make it kind of like little ripples, so to speak. But the data point I wanted to point out, at this rate of decrease, the field would be negligible in about 1,600 years. I'm like, oh, guys, this is pretty bad.
Yeah, what means we ain't got much time left.
And this would suggest it's young. Now, of course, to be fair, they did say, well, it's been average for the last 7,000 years. But I'll point out, well, we didn't have instruments there. That's an inference, and it could be circular inference, which is no good.
Mm-hmm. And we could So what was the what did John Sanford estimate the lifespan on humanity was? I seem to remember a number of about 10,000 years.
Well, this is based on the Bible.
Okay.
But he did properly show that decreasing age. He was trying to illustrate that it followed a decaying exponential.
Mm-hmm.
And
No, I'm saying that like at our rate of mutation, aren't we going to mutate ourselves extinct within like a certain time period?
I don't know if he gave a figure.
Was that Kondrashev's paper? Was that Kondrashev's paper I'm thinking of?
Kondrashev said, "Why aren't we dead 100 times over already?"
Yeah, that's what I was thinking of.
Brian Sykes at Oxford says gives us about 100,000 years. A lot of evolutionary biologists give us about 200,000 years. And I mean, to really be fair, if you think of our genome being 3.3 gigabases, if we're adding 100 deleterious mutations, mostly in the non-coding regions, you'd need about a million years to just get 100 million. So 100 million of 3.3 gigabases. It's not a big fraction. It's not significant. So I don't think I'm we really don't know how much at risk we are. We've lost a lot, but we really don't know a lot. Is all I could say. I wouldn't be too quick to say that we're guaranteed to go extinct by, X date. Just from
right. No, I'm not asking for a date. I'm just more looking for an estimate because I found that was interesting. But yeah.
Already mostly.
Yeah, there's also that.
So Brian Sykes estimated 7,000 generations. That's about what, 20 times 7,000. That's 140,000 years. When he predicts, and he's in like 100,000. He's predicting the Y chromosome will disappear, which is also very interesting because this is an example where you get Mueller's ratchet, where recombination doesn't work, and there may be species out there that there are species that have effectively lost the equivalent of the Y chromosome. So, we don't, it's not looking, the Lord has to return to rescue us. I mean, when I saw this about the geomagnetic field, I'm like, oh my goodness. Even the mainstream's admitting it. And it may be driving climate change. I mean, we could be having if the ozone, NASA is seriously concerned that the ozone layer is being wiped out, and it's correlated to the magnetic field. and they said this could have very adverse effects on our climate. And
Mm-hmm.
I don't think a lot of the climate changes are anthropogenic, natural phenomenon independent of man. And we could be having climate change because, well, I mean, the Lord said, there's the seas will be tossing and, all sorts of, and men's hearts fainting. So a lot of things could be happening. And when I saw that in Wikipedia, I said, boy, it's getting so obvious now. It's even there. So I started to take Russell Humphrey's work even more seriously, and that's why you saw me doing all this math stuff with Maxwell's equations, because I wanted to understand it and then also revisit speed of light stuff. But in any case, you gave me a really good data point on the coelacanths. This is even better than any of the work I did on the coelacanths.
I mean, that's in the evolutionary literature. When they sequenced the genome, it was like 50 to 60% repetitive lineage, repetitive sequencing, and to me that immediately screamed not just a bottleneck, but an ongoing bottleneck, because that bottleneck may still be going for the coelacanth. Because to get that much of the genome to be just repetitive elements tells me that's probably that they're homogenizing their genome really fast,
or they were created recently,
right? Yep.
I think the other side's really starting to fall apart, and we got some diehards like R and Raw, but the people in the know they're in trouble.
But they'll never admit it publicly.
They'll never admit it publicly. Yeah. So I think we're at the point we could shut down our broadcast. I'd like to thank Mark and Godzilla Freak. It's really great to see you guys. Just to let you know, I'm really not trying to just grow my channel. I'm going to be stressed out if my channel grows. Then people will be emailing me and wanting to talk to me. And I mean, I love people, and that's why it's going to be hard for me.
See, that wouldn't be a problem for me, Sal. I just tell people no.
Yeah. See, so
you want me to write your emails for you? No.
Yeah. So especially now that I might be teaching at this new creationist museum and then doing TV shows, and I really think I'm hoping I'll be going on a road show in addition to that. I really won't have. I don't think I have time to deal with first off, the internet drama, and I'm kind of out of that because I'm doing so many nerd things, and that gets rid of a lot of the riffraff. And this is about the right size, for me as far as all the pen pals I ended up creating with this size of a channel. I would be horrified to think what'll happen if my channel grows by a factor of 10, 20, or 30. I couldn't handle it. So let's say I spend 1 hour right now just responding to emails and comments on my videos a week. If this channel grows by a factor of 30, I'm not going to have any time.
So folks, here's what we all need to do. Let's all go out and advertise the show so we can all make Sal's hair grow gray.
It's already growing gray. Yeah. So I want to thank the guys who've been offering moral and financial support. Thank you guys so much. I can't tell you how much that means to me. But one reason I'm having nerd shows is this is a very helpful conversation to my professional work, and I'm really not out to be winning popularity contests. This is that's for other guys, and not for me. So if you could offer a quick prayer, and I'll meet you backstage a little bit, if that's okay.
Sure. Yeah.
All right. Yeah, much love, Matt. Oh, Matt, you're still here. Okay. All right.
All right, guys. Thanks for tuning up. Appreciate it. And hopefully we gave you some things to think about. I know we probably all bored you to death. There's also that. But thanks for tuning out. Dear God, our Heavenly Father, I humbly come before you, praising and glorifying you for the day you've given to us. Thank you for the opportunity I had to hang out with Sal and nerd out about some science. Lord, I pray that you will just help what we've said to be beneficial to the people who have listened. And Lord, I pray also that you will give these good people here who are supporting Sal, some benefit from what we've said. Lord, I pray that you will help them to understand it. I know we talked in a little bit of nerd speak, but I pray that you will just help it make it clear and understandable for anyone else who listens later. And Lord, I pray also that you will help grow Sal's channel so that he has to spend more than an hour a week responding to emails and comments. And that you will just grant him wisdom as he pursues his career, and you will help him bring glory and honor to you through what he says and does. Lord, I pray that you will give us all a peaceful night's rest tonight so that we can be refreshed and ready to serve you in our churches tomorrow morning. I ask this in the name of your son, Jesus. Amen.
Amen. All right, guys. God bless you. Take care. Have a blessed Lord's Day.